Hello and welcome to Transcription. This is the first in a two-video series on the central dogma of biology. The central dogma is the idea that DNA, which is the genetic material, serves as a template to make RNA, which will then serve as a template to make protein. We call the process from DNA to RNA transcription because you're going from the language of nucleotides in DNA to also the language of nucleotides in RNA, and although they are slightly different, they kind of follow the same principles, so we like to think of it as a rewriting or a transcription. The second part of this process we call translation because you're going from the language of nucleotides to the language of amino acids, which is like a whole different thing, as we will soon see. The two figures here, the two cells, kind of show an overview of what is happening throughout both these processes, transcription and translation, in a prokaryotic on the right versus a eukaryotic on the left cell. In the eukaryotic cell, we have a nucleus, which makes things a bit different. So DNA is stored in the nucleus, and there it will get transcribed into RNA. It'll get modified a little bit. It'll get exported from the nucleus, and then in other places in the cell, the cytoplasm and the rough endoplasmic reticulum, it will be turned to protein or translated to protein with ribosomes. In the prokaryotic cell, we don't have a nucleus, but we do have DNA that sits in an area called the nucleoid, and the DNA will be transcribed into RNA, which will be translated into protein, kind of all at once, and it can even happen simultaneously where the RNA is not even finished and the protein is starting to already be made by the ribosome. I also want to clear up something that I just misspoke about just a couple seconds ago, where I said something along the lines of RNA is turned into protein. These molecules are not turned into each other. For example, the DNA is not going to be modified chemically in order to turn into RNA. The DNA is going to serve as a template, and the RNA is going to be made based on the sequence of the DNA. And same for the protein. The RNA does not turn into protein. It's only going to be used as a template in the act of making protein. A really important aspect of this is called the genetic code. And the genetic code is how we take every 3 nucleotides in DNA or RNA, and we can translate those into one amino acid in a protein. So I can take a DNA sequence or an RNA sequence and predict what protein will come out of it in the end. This particular table here shows for RNA every 3 nucleotides, and 3 nucleotides in this case we call a codon. One codon is going to make one amino acid, or it can send a couple other messages. For example, here you can see that AUG does not code for an amino acid. It tells us to start. And these 3 sequences do not code for an amino acid. They tell us to stop translation. So how do we read this crazy table? Well, we're going to look for our first nucleotide here. So let's say that my RNA was AUA for this particular codon. I would find A here, and then here where it says second mRNA base, I would find U, which is here, and where those two intersect, there is a series of 4 codons, and then we find AUA, and we can see that that is going to code for isoleucine, which is an amino acid. An important thing about this genetic code, or 2 important things actually, are that 1, it is redundant. So what redundant means is that there are many codons that code for the same amino acid. For example, all these 4 codons code for leucine. Another important thing is that it's non-ambiguous. What we mean when we say non-ambiguous is that if you have a codon, it can only code for one amino acid. For example, AUA definitively coded for isoleucine when we looked. It could not code for leucine, it could not code for alanine, it could not code for anything else. This is non-ambiguous, or it's not vague, it's very specific. Another thing that's important about the genetic code is that it's universal. So look at these pictures here. This shows something pretty cool, which is that this genetic code can work in nearly every organism, and I'm sure every organism you'll encounter in your biology studies, in the same way. For example, here we're going to see in yellow the tobacco plant expressing a firefly gene, or this pig can express a jellyfish gene. So people have done some really cool stuff with this, and we've actually also been able to do a lot of useful stuff. For example, we can take a protein that people need to take, for example, insulin. Diabetics need to take insulin, and we can actually make bacteria overexpress that protein and harvest it from bacteria. I think that's really cool. Anyways, so here's a more detailed view of what exactly is going on in transcription and translation. So we're going to take a double-stranded DNA, and we are going to make out of it one single-stranded RNA. So this double-stranded DNA, we can kind of name it some different things. So you can see here that this strand, this upper strand of the DNA, is called the template strand. It's called the template strand because it serves as a template for the RNA. So the base pairing between the DNA and the RNA is actually happening between the RNA and the template strand. You see how it's U and A, and G and C, and G and C. These are the ones that base pair. If you try to base pair the RNA with the other DNA strand, it will not work. The other DNA strand we call the non-template strand. I know, very creative naming. Another name for these two DNA strands, or another set of names, are calling the template strand the antisense strand, and this one the sense strand. Now, why would the template be the antisense and the other one be the sense strand? Well, if you look at the sense strand, aka the non-template strand, that's the one that kind of looks like the RNA. The only difference is that T's are replaced by U's. So the sense strand is called the sense strand because it makes sense. I can take the sense strand, replace all my T's with U's, and then use that genetic code to figure out what kind of a protein that I'm getting. A third set of nomenclature for these two DNA strands says that the sense or non-template strand can also be called the coding strand, for basically the same reason that we said it was called the sense strand. And then the other one, I've never really heard it be called non-coding, I've just heard template or antisense. So we're going to use that DNA, the template strand, to make an RNA, and the RNA, every 3 bases, are going to be used to make one amino acid. So you can see the sequence here, it goes tryptophan, phenylalanine, etc., etc. And now that I've given you an overview of transcription and translation, I'd like to zero in on transcription and talk a bit more about what exactly is happening as we turn DNA into RNA. In transcription, we have 3 phases we go through. They're called initiation, elongation, and termination. In initiation, we're going to be starting transcription. And we start transcription by binding an enzyme called RNA polymerase to DNA. RNA polymerase is going to be the enzyme that makes a polymer of RNA. So, how does it know to bind to DNA? It's because before every gene, there is a sequence of DNA called a promoter, and the promoter can bind RNA polymerase. Now, a lot of promoters sometimes share DNA sequences with each other because apparently there's like a kind of a set of DNA sequences that work in terms of binding RNA polymerase. And if many different organisms or many different genes kind of share a DNA sequence, in the promoter or in anywhere else really, we call it conserved. So that just means it must be important because everyone has it. One of these conserved or consensus sequences is called the Tata box. So this is something that we often see in the promoter, and it's called the Tata box because literally it makes a box of TATA, ATAT. So that's one sequence that might be important to know depending on what biology class or what test you're studying for. After initiation, where RNA polymerase binds, we're going to have elongation, where basically we just elongate or keep making the RNA strand. After elongation, which is relatively a boring step, we're going to get to termination. In termination, the RNA polymerase will reach a sequence where it knows to dissociate from the DNA, and transcription will be complete. In prokaryotes, sometimes, like I said, before transcription even is terminated, translation can start by binding of the ribosome to the RNA. But in eukaryotes, this cannot happen because translation occurs outside the nucleus and transcription occurs inside the nucleus. Another aspect of transcriptional initiation has to do with a set of proteins called transcription factors. Transcription factors are proteins that can either say, hey, RNA polymerase, get over here, or they can say, RNA polymerase, we don't need you right now. It depends on the transcription factor, and unless somebody tells you in whichever particular test question, you can't really assume that a transcription factor is activating a gene or silencing it. In eukaryotes, we have these transcription factors. Prokaryotes actually do not have transcription factors. Prokaryotes do, however, have an analogous or similar set of proteins, and we call them sigma factors. But this distinction is important because often questions will make use of it, and like if you're answering a question on prokaryotes, the answer that says transcription factors will be wrong. So on the right, you can see a schematic showing how transcription factors work. They're going to bind to DNA, and what's not shown is that they're going to kind of change the shape of how DNA is folded up, and they're going to make a shape that is more conducive to binding of RNA polymerase. One more thing I want to go over with the process of transcription itself is how the DNA strands and the RNA strand fit together during elongation, and we kind of already talked about this, but if you look at these three strands of nucleotides in this figure, then you can kind of see where the RNA is, and you can see where the RNA base pairs with a DNA strand. So the template DNA strand is here at the bottom, and here is where we are base pairing. So this U will come in here. This U is paired to this A. Here we have base pairing. Here we have base pairing. Here, and so on. And then the other strand just kind of sits there, but the other strand, the non-template strand, is the one that kind of looks like the RNA. So you can see like this U is kind of analogous to this T, this U and this T, A, etc., etc. So what's happening here also is that the RNA is going to be made from 5' to 3', just like any nucleotide is made from 5' to 3'. But that means that we are actually seeing our template strand go from 3' to 5'. So the template strand is this one, which starts at 3' and ends at 5'. The non-template strand will go in the same direction in terms of 5' and 3' as the messenger RNA. So once we've terminated transcription, it's not actually a messenger RNA yet. We like to call it pre-mRNA or pre-messenger RNA. And in order for the pre-mRNA to become a mature mRNA, we need to modify it in 3 special ways. Oh, and also this is only in eukaryotes because remember, in prokaryotes, transcription can literally happen at the same time as translation. So these 3 modifications I'm about to talk about are unique to eukaryotes only. So in eukaryotes, we will see what's called a 5' cap. And this 5' cap is just a modified G nucleotide on the 5' end of the RNA. It's called 7-methylguanosine. And this does not mean that you have a methyl at position 7. It means you have 7 methyls on the guanosine. I don't know if you care, but when I learned about this, I asked that question, and I remember getting answered that way. But anyways, what this cap serves to do is to protect the RNA from all the RNases that are outside of the nucleus in the cytoplasm. Because the cytoplasm is a scary world, and the RNA has to go out there to be translated into protein. Eventually, we're going to export this mature mRNA, once it's mature, out of the nucleus. And there are RNases there to degrade RNA. Now, why the heck would our cell want to have RNases floating around? Well, one reason why it was useful for us to selectively decide via transcription factors which genes should be on and off at a given time is because there are some genes that we only want on some of the time. For example, let's say that I have a gene that promotes mitosis. So whatever protein this is, and if you've learned about mitosis, you've learned about proteins like this called cyclins or cyclin-dependent kinases. What if my protein promotes cell division? Well, I can transcribe it and translate it, but if my RNA stays around in the cell forever and I keep translating my gene, even when I want it to be turned off, my cell's going to be dividing all the time. And you know what we call that? Cancer. So the moral of the story is that we want our RNAs to be temporary because we want to have a way to turn gene expression on and off. So we are going to have RNases in our cytoplasm to degrade our RNAs after a while. We want them to have like a shelf life, and then we want them to expire or die. But we do want them to stay around long enough to be translated, which is why we build in protections like the 7-methylguanosine cap. The reason why that works is because an enzyme, an RNase, will see the RNA, be like, oh, look at this, I'm going to destroy an RNA. And then they'll look at the 7-methylguanosine cap and be like, what the heck is this? This isn't a nucleotide. Another modification is called the poly-A tail. And the poly-A tail is just what it sounds like. It's at the 3' or tail end of the RNA. We have many A's, actually like 100 or 200 A nucleotides. And this also works to protect the mRNA from degradation, because an RNase will come along and be like, ha ha ha, I can degrade RNA sequences that are important in protein making, and it'll cut at the poly-A tail, and the mRNA will be like, psych, that wasn't important, right? The third modification is called RNA splicing, and I'll talk more about this in just a minute. But the bottom line is that these 3 modifications in eukaryotes serve to protect the RNA from degradation in the cytoplasm for a short while, and also they're going to help the RNA know that it has to get exported. It's going to help to signal exportation from the nucleus. So how does this work? Well, you can see in this diagram the cap and the tail. And then in the bottom diagram here, it's showing RNA splicing. So RNA splicing has to do with the idea that a lot of genes contain what we call introns, or intervening sequences, that we don't want in our protein. So there is actually a complex called the spliceosome, I know, very creative name, that's going to take this RNA, this pre-mRNA, and it's going to cut out the introns and leave sequences that we call the exons. So here you can see in dark red are the exons that we want to keep, and we end up keeping them. In light red are the introns, and there can be more than one intron. This just shows one. And the spliceosome, which is a complex of small nuclear RNA, which is a non-coding RNA, and small nuclear ribonucleoproteins, which some people call SNRPs, a really ugly name, I think, are going to contribute to the spliceosome, and they're going to basically make the introns into a loop. So the intron will be in a loop, which sometimes people call it a lariat loop, because a lariat is a lasso, and when people discovered this, they thought it looked like a lasso. And they're going to bring these two ends together and cut, and this lariat loop will come out, and we will be left with a mature mRNA that only has the exons in it. Now, what the heck could be the evolutionary use of having random intervening sequences in our RNA? Well, this is kind of twofold. So first, if you have intervening sequences in your RNA that don't really serve any function yet, it's an opportunity to have mutations that don't really put you at risk. And if you have opportunities to mutate, you have opportunities to get to have an advantageous trait and become a better organism. The other use that splicing has is to make multiple isoforms of a protein. Isoforms are just different but similar forms of a protein that have maybe different amino acid sequences in them, but kind of serve the same function or have the same shape overall. So we can actually do this thing called alternative splicing, where the exons become introns, the introns become exons, and we can basically pick different stretches of nucleotides to put in our protein and make different isoforms of our protein. So for example, there are different isoforms of hemoglobin, like fetal hemoglobin, that actually has a higher affinity for carrying oxygen than regular hemoglobin. But anyways, once we modified with the cap, the tail, and the RNA splicing, for eukaryotes at least, we can export from the nucleus and get this RNA translated, which you can look at in the next video translation.